So welcome everybody, and it's my pleasure today to welcome you to the seminar series in quantitative life sciences and medicine. And we have the pleasure of hosting today Dr. Dana Peer, the chair and professor in computational and systems biology at Sloan Kettering Institute. She holds a Howard Hughes award, and she was previously at Columbia. And she's looking at genetic variations, single cell, regulatory networks, cancer, all kinds of questions about cell circuitry and tumor immune ecosystems. So she's also on the editorial board of Cell, and she's won a number of other awards in addition to the Howard Hughes. She's had a Burroughs Wellcome Fund Career Award, an NSF Career Award, Stand Up to Cancer Research Grant. And she has her PhD in computer science from the Hebrew University of Jerusalem, and has a background in machine learning and Bayesian networks as well as cancer. So we're really looking forward to her presentation here today. And please go ahead.
Thank you very much for the introduction. If we can summarize the introduction as I use compute and data to ask fun biological questions with colleagues who are fun to work with. That is a much more honest description of my research, but there has been a lot of focus on cancer development, cell decision, and tumor immune interactions. And here, this is set up to go forward on its own in some stupid way. Sorry, let me turn off auto timings. Set up show manually, no timing, sorry. Anyways, of course, my disclosure, I'm on the advisory board of In Vitro. That's the only thing I've ever agreed to do, because saying no to Daphne Kohler is impossible, but nothing in this talk relates to that in any way. So you're only going to get my opinionated view of things, but with no financial interests. And so recently, one of the things that just fascinates me the most about biology is the concept of cellular plasticity, that the cell senses its environment and can
Dana,
actually
we don't see your slides.
Oh, it hasn't been properly shared. All right, sorry. I thought I did a new share slide after killing that thing. All right, now.
There we go. There we go.
This is a pretty, thanks for letting me know. So this is a new, so this plasticity exists in normal cells, but is really amplified in cancer. And it's not, genetic mutations, but rather this remarkable plasticity, which is driven by mutations originally, that enables the cell to do all its nastiest business, such as metastasize or develop drug resistance. Now, the reason I personally love single cell data, and the reason that I just suddenly became like single cell everything, and I don't think I have any projects that don't have to do with single cell data, is because when you take single cell data from a tissue or even from a cell line, you reveal a continuum of cell states. So rather than needing a time series, you can actually get your full time series from one single sample, because the cells are asynchronous and you can see things in all potential time points. Thus, you can really infer a trajectory and get dynamics from one sample. So one sample often has many, many thousands of cells, which again gives you the statistical power to use machine learning and computational techniques to actually say something interesting. So I'm going to tell you, I'm going to start with a little bit of sort of computational toolbox, so we'll be all on the same page. And after this computational toolbox, we'll see how we can use it to ask interesting and fun biological questions, and throw in some more computational tools along the way. So we're going to begin with one of my favorite methods, Palantir, which was developed by Manu, who's now his own faculty member at Fred Hutch. And the idea is that pseudo time is very well accepted, and there are hundreds of algorithms for pseudo times, but one of our points in Palantir is not only do cell states reside along a continuum, but actually cell decision. There's no point of decision, but the cell decision is also of probabilistic nature when a cell gradually over the course of the trajectory leans towards one or another fate, and we wanted to compute that. So pseudo time, the basic idea of really modeling cell fates and capturing this continuum that I've been talking about. This is probably known, but I just want to mention it, is really the main fundamental object in single cell analysis is the neighbor graph, where every node in the graph represents a cell, which is connected to its most phenotypically similar cells. And we actually can stay within phenotypic space and can stay and do all our computation within states where there are actually cell phenotypes by paths and traversing this graph, really learning the global structure of the phenotypic manifold, where phenotypes really, you can't have any combination of gene expression. There's a real geometry to the states of possible cell states. Now, here we said, okay, let's take this neighbor graph and direct it by one of many ways. Here we used simple pseudo time, starting from an initial stem-like or progenitor cell. And once we have a directed graph, then we have all the tools of Markov chains to our advantage. And Markov chains just have, it's a huge and powerful toolbox. And basically our approach was, let's model development as a Markov chain along this directed neighbor graph. And therefore, for each cell, using sort of this Markov chain, we can infer what are its possible cell fates by computing all possible paths and all possible probabilities. So if we want to know what terminal fate this red cell will reach, we need to compute all paths, the probability for each path to get to the red, the blue, and the green. Now, that's millions of paths. And again, I said, I love Markov chains. And the nice thing is you can easily take a graph, turn it into a cell by cell matrix, where each entry in the matrix is the probability in the original graph of transitioning from cell A to cell B. But then in order to compute longer and longer paths, all you need to do is power the matrix, simple linear algebra. Here's all the paths of length 5 and where the cells can get, as well as which cells can reach each of these terminal states with what probability. And you can see that as the cells can traverse longer and longer paths, they have a probability distribution of getting farther and farther in phenotypic space. And there's this beautiful closed form solution that allows us in a quick and closed form solution to compute for each cell the probability of reaching each of the many possible terminal fates. This is a very powerful tool, and we've been using it in a lot of biological applications. And one of the most powerful and my favorite is a collaboration with Kant and Tanaka's on early development of the endoderm, the layer that's going to give rise to all our internal organs, the gut, the lung, the pancreas, liver. And this again was a close collaboration with Manu on the computational side, and Sonia, a truly amazing experimental biologist in Kat's lab. And so the full paper is again published so you could read about it. But again, I just wanted to highlight the power of Palantir and the concept of plasticity. So here, basically, we have the first 3 days of development, where marked here as embryonic days. And we ran Palantir on this. We saw, well, first of all, the force directed layout gave a bridge, which everyone thought was noise at first. But Palantir actually inferred that this bridge was real, that in addition to this initial point, at the first day, point 3.5, where the cells decide if they're going to become epiblast, embryonic, to form our organs, or if they're going to become extra embryonic to define the yolk sac and the placenta and the umbilical cord. After this initial decision, these epiblast cells, these embryonic cells, have a state that's actually, again, highly plastic. And there's actually a bridge for embryonic cells to extra embryonic cells at day 5.5. And we did a lot of computation to convince our collaborator that this is real and this is robust. And we finally convinced her to do lineage tracing. And lineage tracing with 2 different systems showed that these embryonic cells that are just, set to become embryonic, very, very different transcriptionally and phenotypically, are able to migrate to the membrane and to change their fate and find markers and morphology of extra embryonic fates. This lineage transformation actually happens again at day 8.75 during gastrulation. Here you see an embryo gastrulating. It's an image again developed by Kat, and I think this is just like an incredible image where you see the extra embryonic cells being actually the primitive endoderm cells and the extraembryonic cells being in red, being sucked in. So basically these green cells were supposed to be extraembryonic, but as this sort of thing turns inside out to form the gut tube, some of these green extraembryonic cells just physically get sucked in. And this was observed by Kat more than a decade ago, but she wanted to know, okay, what are the fate of these extraembryonic cells that get physically sucked into the embryonic gut tube? With single cell RNA-seq, we could look at it, and at this time point, rather than pseudotime, the strongest diffusion component, the strongest nonlinear component of variation in this data was pseudospace, and the data organized along an anterior from the head to posterior axis in the tail. And while the cells are morphologically identical along a smooth gut tube, at this point, you could see that the cells clearly clustered into very clear clusters that each represented the promotal organ that would bud out of that location along the anterior posterior axis, expressing many of the key lineage regulators and many of the key markers of each of these internal organs. And we could see that the major transcription factor that drives each of these organs, depending on where it is along this pseudospace, is expressed both in the embryonic and the extraembryonic cells. So both the embryonic and extraembryonic cells, we had lineage tracing so we could know their origin, both expressed the master regulators. And in fact, controls for where they are along this pseudospace, they reached 99% correlation, even more than 99% correlation. There were about 6 genes that were expressed only in the extraembryonic cells, some early memory. And this really shows remarkable plasticity, that while the cells are really, they know in advance where they're going, they have internal transcriptional programs to go one way. If they find themselves in a new environment, they can suddenly sense that there's a change in this environment and completely look at their neighbors and say, hey, all my neighbors are going to become liver. I'm going to make this dramatic plastic change of my fate and become a liver as well. And I find this really remarkable. Now this plasticity is really further amplified to crazy degrees in cancer. And this is one example, again, a published paper where you could look at where we really showed that metastasis is really an organ that basically using programs of organogenesis, early lung organogenesis, to try and grow a lung at the wrong time in the wrong place. Really,
can I interrupt?
What?
Sorry,
Yeah.
I have a question coming into the chat, if you don't mind. If the transition matrix can be interpreted as a probability of transitions between cell states, can we say that the region under the diagonal represents reverse transitions?
So again, there's a lot of assumptions that are made here. And, in development, actually, the assumption that, largely development goes one way from more immature to mature cells. This is, of course, broken in regeneration and cancer. And if you give me one more slide, I'll actually address what we do when that's broken. But generally speaking, our assumption when looking at development is that development is more or less one way. Cells can go a little bit back, which capture the mistake in our neighbor graph, but development is typically one way. That's probably an oversimplification, but as a first order approximation to give us some learnable structure that works. Anyway, just to say here, we look at the metastasis cells, and this is, samples taken from the clinic across the street at Memorial Sloan Kettering. And we could really see the lung organogenesis program following the same order with the same transcription factors and the same downstream programs. And, as I promised, like someone asked the right question, we make the strong assumption about, one directional ordering in development, but this is complete, and this is an assumption made by almost all, if not all, pseudotime approaches, that they assume starting point and a forward direction. This assumption is clearly broken in regeneration and cancer. And so the nice thing about the Markov framework is that we can really use different metrics to develop the similarity, for example, epigenetic marks. And we can change the way edges are oriented. And I'm going to show you one example where we use RNA velocity, which looks at the ratio between introns and exons to sort of assume that the introns are made first and then the exons later, and you can sort of see where transcription is going from this intron to extron ratio. Of course, this is a ridiculously noisy measure, but it gives you some idea of directionality, which you can incorporate in the graph. And this is exactly what we do with CellRank, which is a joint collaboration with Fabian Theis and Marius, who's in the lab. And I think somehow the slides got switched in order. But we're really going to marry this velocity with the sort of Markov graph and Palantir framework by saying, let's take each cell in the phenotypic manifold. Let's look at the velocity as it's computed. That sort of has one direction, imperfect and noisy. We can look at the neighbor graph and really, rather than just use some simple pseudo-time assumption of immature to mature, we can say cells are more likely to transition in directions that best align with the velocity. Again, this is probabilistic, so you can go any which way, but if you're better aligned with the velocity vector here, you have a higher probability. And this allows you to go back and forth and use some type of information from the data to sort of orient your cells without making strong assumptions. Now, the nice thing here is that it takes these very noisy velocities, infuses some, observed, measured metric of direction, but it really constrains the transitions to remain within the phenotypic manifold, to remain within observed and possible cell types, which is not always the case for velocities that sometimes point in directions where there's no possible cell state. And these very, very noisy velocities sort of propagate over this phenotypic manifold through the Markov field and really create robustness through global properties. Now, of course, I said the nice thing about Markov networks is that they're awesome, and there's a lot of math and there's a lot of tools that you could use. And here we can actually cluster them, cluster the dynamics themselves, infer automatically initial states based on these dynamics, ending states, intermediate states. It's really taking these sort of individual cell dynamics and coarse graining them using a method that was actually originally developed for protein dynamics. And the ability to automatically infer these initial states rather than feed them in through prior knowledge is critical when such initial states aren't necessarily known. And here we demonstrate that and velocity and CellRank is on bioRxiv and soon to be in Nature Methods. Since I said, okay, the true test of the method is regeneration, here's one example in regeneration where CellRank infers the opposite direction from the far more mature goblet cell to the far more immature basal cell. So this is going in the opposite direction of maturity, the opposite direction of regular development. And in fact, there's no direct path known between goblet and basal cells. It's usually basal, club, and goblet. And here we infer a reverse dedifferentiation of a goblet to a basal cell. Now, our lineage tracing sort of had some technical problems, but we could use imaging over time series to show this intermediate states exactly as we predicted them and how they properly accumulate over time, really showing imaging and time series support for our transition at the single cell layer and spatial support, because this is also spatially organized from this more mature goblet cell into the far more immature basal cell with all the inferred intermediates along a spatial axis here in between. Okay, so these are just a bunch of tools into my toolkit that I just wanted to share with you so we could actually use them to ask some meaningful biological questions about cancer. And this is a great collaboration with Scott, who's down the hall. So we're all on the same floor. So there's a lot of collaborations that just happen with people sharing lunch. And this is one that started between Cassandra, an amazing graduate student in my lab, and Dorena, a postdoc in Scott's lab. And again, the best collaborations are when the experimentalist and the computational biologist are really working hand in hand daily and communicating. The gut endoderm work was like this as well. And here we really wanted to sort of understand how why injury increases pancreas cancer. Both in pancreas cancer, pancreatitis is often leads to pancreas cancer, and likewise a lot of inflammatory diseases lead to colon cancer. So here, most cancers are really, most studies of tumor genomics is really mutational focused. But of course, I've made it clear that I like the good functional stuff that isn't directly coded in mutations. And particularly for this project, we wanted to know what happens between the initial driver mutation, in this case, KRAS, oncogenic KRAS, and a later driver mutation such as P53, where there's a lot of stuff that happens in this pre-malignant stage that's poorly understood and not believed to be driven by mutations. It is believed to be driven by microenvironment, as I said, tissue context, injury, inflammation, and of course, if it's not genetic, then it's epigenetic, and we wanted to look at all of that. And since almost all studies are profiled here at these nasty terminal, late stage, malignant states, we actually wanted to use the powers of mouse models and inducible mouse models to really study these understudied and super interesting early states. So one of the things the Low Lab is known for are these super powerful mouse models. And here we compared normal regeneration following injury, where the cell, which is completely reversible, and the cells get injured, turn state, and regenerate, to a mouse model with oncogenic KRAS just in the acinar pancreas cells. So this is a small population in the pancreas believed to be the origin of pancreas cancer. And we have the KRAS mutations just in one subpopulation in the pancreas. And also these cells are labeled for sorting so we can enrich for this rare epithelial populations to get lots of cells and good stuff, as well as a later stage where we have a loss of heterozygosity of P53 to sort of push the cancer into full malignancy. And we took all these time points and notice these color codes: early injury, late injury, but still pre-malignant, and fully malignant state. And we'll use these colors later. Of course, at first you want to sort of see your data, whether the data is good. And I'm really pleased with this data because it's some of the highest quality data that I ever saw. Here we look at the sort of really rare subtypes, and what you see here is that cells from different samples and different even malignant, pre-malignant, non-injured states all overlap these sort of normal cells with no sample-to-sample alignment. I mean, the replicates here were perfect. We see that these three separate malignant tumors actually do take completely different phenotypes, but because of the overlap of the normal states and the overlap of the replicates, we really believe that the difference is biological. And in fact, here we see a shared state that's shared by all malignant samples, both metastasis and the three malignant tumors. If we take this diffusion component, the main component of variation, strongest axis of variation in our data, it really matches this progression from regeneration to the left, to super malignancy, to full on malignancy on the right, and all these earlier intermediate states in between. And I always like comparing to sort of the clinic, which we have access to, because mouse models are great and perturbable, and you can study all sorts of things, but you always want to see that the trends that you see and are excited about in the mouse models really are also recapitulated in the clinical samples. So now we're going to really zoom in to this sort of KRAS, oncogenic KRAS mutant epithelial things. These are just the epithelial sites because we had them, with a marker so we could sort and enrich for them. And here I'm using a force-directed layout, which really better represents the diffusion components and is a better way to sort of see progressions. And the force-directed layout really organized the data into the sort of pre-malignant component to the malignant component on the right. So if we really focus on the early states without injury, and 1 or 2 days after injury, we see incredible heterogeneity, much more than the heterogeneity that we see in the wild type. But of course, this is a mess. This is regeneration. We don't know where the starting cells, what is the direction of transition. These are all new states, and it's really hard to say anything about directionality. So cell rank to the rescue, and cell rank really predicted 3 different origin states. And actually, this really aligned a lot of arguments in the field, because a lot of people said, my marker NES plus is the origin. No, my markers are the origin. And the answer is there are multiple origins, and actually it also aligned multiple markers per origin. So now we can map the origin. Now I want to state that the reason I could run cell rank, which is based on velocity here, is that I was only looking at 0, 1, and 2 days after injury. Velocity should really only be used in short-term changes. Velocity really captures the 8 to 12 hour range, and trying to use velocity on these advanced cancers and connect weeks apart with velocity just doesn't work. This is very short-term transcriptional dynamics, but this worked because we were, really focused on this very early time point. Now we could see what happens during injury, and we see that during acute injury, the cells are pushed towards a more PDAC-like state, even with this bridge that really pushes them almost towards PDAC, and there's a break, so they don't quite cross this bridge. This break happens to be P53. And while I'm showing you this and explaining to this on a 2D image, because I can't show you a multidimensional nearest neighbor graph, when I talk about distances, I always test them in the high dimensional space, because any projection in 2 dimensions can be very misleading. But what happens is when the inflammation clears, and when we look at the precursor lesions 3 and 5 weeks later, the signal reverts back, and they're actually farther from the malignant state. Now, from a previous paper that we did together, we know that chromatin is playing a really, really big role in everything that we're looking at. So we decided to focus and analyze the chromatin data. First, we decided to look at bulk, and the bulk chromatin data across the stages and across the replicates really confirmed that this early injury state, 2 days after injury, seems to be in between a Panin route that's sort of more benign and a nasty adenocarcinoma route. And we could sort of map the open and closed peaks in each of these samples to the single cell data. And we indeed see that this injury, these blue cells that are 2 days after injury, have open chromatin that we observe both in Panins and open chromatin that is typically only observed in advanced PDAC here in purple. So this is really interesting that these blue cells seem to be very, very, again, my favorite word, plastic. And we decided to directly measure and observe this plasticity. So here we decided to build a very, very simple classifier. And we used the expression of each one of these states, early Panins, PDAC, metastasis, to try and find a gene expression classifier that will tell us which state it's in. The transcriptional signal is so strong, this is an easy 5-year-old classification problem, and it's very, very easy to get a classifier that's very accurate. The goal was to use the classifier developed on these late stages and apply it to these more poorly understood early stages. And basically using the classifier that we developed to classify late stages, we applied the very, very same classification to early stages, either with no injury or with 1 or 2 days after injury. And we found that, many KRAS mutant states are really primed in advance in their epigenetics towards the benign states. Injury really pushes many more cells and shifts them towards a higher likelihood for a PDAC-like state. But there's a lot of intermediate cells that are in between, that are plastic, that can go either way, an inflection point. And indeed, this inflection point, these probabilities that are very plastic and unclear directly map to these points that cell rank pointed out as our initial probabilities, really showing that these initial starting points are plastic before they make their choices of which way to go. So we decided, okay, we've actually juiced bulk ATAC-seq pretty well, but then came out 10x kit, so we could do single cell ATAC-seq and look at the data at the single cell level. And I don't want to go too much into our computation here, but just to say that we recapitulated many of the same populations we saw in the single cell RNA-seq, as well as this sort of plastic priming towards benign, malignant adenocarcinoma and in between. We like this idea of classification, so we wanted to ask, okay, can we predict based on a tax-seq what cells will be doing? And again, we build a classifier for each of the many, many cell states. Now we're not looking at big classifications, but actually we finally clustered the data, found different clusters in our data, learned a classifier based on transcription, what cluster each cell belongs to, and said, okay, if we look at the open chromatin as a proxy for the activity of each gene, in some places, the open chromatin of the red gene, the open chromatin of red genes was really clearly stated. This cell is expressing red cell genes, other times green cell genes, but we also had these plastic states of open chromatin of two different cell types. So we had many of these classifications as, mixed as a probability to be either red or blue. So a tax-seq was a lot more plastic than the RNA-seq. And looking at the classification confusion matrix, we saw that there's a lot of transcriptional states which actually don't have high probability in any of the observed epigenetic clusters. And these are clearly not explained by chromatin dynamics. We assume that it's the microenvironment that is largely driving these states. But more interesting, there are a bunch of epigenetic clusters right here on the bottom that have high probability to be in a number of different transcriptional states. So it's not a one-to-one mapping like up here, but actually a one-to-many mapping of an epigenetic cluster that can result in a number of different epigenetic states. And in fact, if we ask, okay, what are the most plastic states? There again, these more progenitor states, these states that are early on in our cell rank model. Because we have the full transcriptional profile for each of these states, we can see what are the other pathways and processes that are most enriched in these most plastic cells. And at the very top, like in a huge degree, by a huge margin, not cherry picked, but number one with the margin was cell-cell interactions. So cell-cell interactions and a lot of activity in cytokine receptors, and really correlated entirely with the degree of plasticity of the cell. And when we actually looked at other components, specifically the immune component, so we profiled the epithelial compartment, but we also profiled the immune compartment. And I want to remind you that the immune compartment is completely wild type. It's interacting with the KRAS mutant epithelial cell, but this completely wild type cells dramatically remodeled, even more so than the epithelial cells. And a lot of this remodeling involved lots of changes in ligand and receptors. So we decided to focus our analysis just on ligand and receptors. And here we actually saw that the ligands and receptors really organized into modules. And you could see these clear sets, and this is a sort of gene-gene co-expression matrix where every column or row is a gene, and it's really red if they tend to be co-expressed in the same cells. And we see a lot of diagonal action, but we see a lot of red, a lot of off-diagonal action. So we decided to use an algorithm that doesn't just cluster, but allows each gene to participate in multiple modules. And we found a number of what we called communication modules of tightly co-expressed receptors and ligands. We found a total of 6 of these. And if you plot, and these are sort of gene programs, and we can say how active is each gene program in each epithelial cell. And we see that these gene programs really map to the different clusters and different compartments of the epithelial, really defining cells that define sort of 6 different behaviors of our mutated epithelial compartment. Now, these modules are components of both ligands and receptors, where you have a receptor and a ligand co-expressed in the same cell. So it really defines the mode of communication. So we decided to, before I just want to forgot to mention this really important thing. Everything that you see here, all this crazy activity, all these crazy open and expressed receptors and ligands, super quiet in wild type. Wild type is very quiet. It doesn't have all this activity. And in fact, even this sort of pinkish activity that you see here, these are actually KRAS high cells during the injury, where KRAS gets temporarily activated and then they express it. So it really shows that KRAS is driving the opening of this communication. But in wild type, most of the cells remain quiet and a small subset of cells transiently open up to this communication, which is shut down afterwards. Whereas here, these are constitutively open. So we could build a network of potential interactions between receptors and ligands. And what's super important to stress here is we built this network based on interactions that are only possible in a wild type setting. So either the receptor or the ligand in each of these connections has to be completely off in the wild type setting. And so these are interactions that are only possible in oncogenic KRAS. And look at how many connections there are. And the number of connections and the size of the module is sort of drawn here. So I have these master modules, and these same master modules are actually also expressed in the more malignant PDAC in the later purple stages, especially in this sort of shared PDAC state that's shared by all of them. And in fact, we observe these very same modules in human PDAC samples, as well as in human colon cancers, by the way. And so now we really wanted to dig into this crosstalk. I really hate these fuzzballs, so I like looking at everything. I love matrices, if people didn't notice, and we can have a sending score and a receiving score for each cell type in terms of how many messages it can send and receive. And we see our master regulators, the gastric and the progenitor modules. We could look, okay, I mean, behind this is real genes. They're real receptors and real cytokines. We could look at who they are. And these are enriched for the very, very same genes that are considered high in these high plasticity associated genes. So these are the very same genes. These red genes are high in high plasticity cells. And we noticed IL-33, which, computation is nice, but sometimes a biologist that knows something helps a lot. And we know that IL-33 is really important from Dorena's previous paper. So we were very happy to find it, because IL-33 alone mimics injury. If you just throw IL-33, you can really mimic the effect of injury. So we decided to focus, what is the communication that is possible by IL-33? Sort of built an IL-33 centric network. We saw that IL-33 is expressed in these epithelial gastric modules. These are received by Tregs and ILCs in the immune compartment, the wild type immune compartment. These very same cells are also expressing IL-4. And once this IL-4 is activated, they can communicate back to the gastric module, the progenitor module, as well as to some macrophages. And I wanted to, again, highlight that these two populations are super active also in the shared, more advanced PDAC cluster. And again, to highlight the difference between wild type, IL-33 is absolutely not expressed in wild type epithelial. IL-4 is absolutely not expressed in wild type epithelial. So what we really have is that active KRAS initiates a very powerful feedback loop that dramatically remodels both the immune and the epithelial compartment within 24 to 48 hours. So we want to see what happens when we knock IL-33 down, since this loop is really driven and starts out with IL-33 in the KRAS mutant compartment. Now we used Milo, which is it was developed by John Marioni, and it's my favorite algorithm to compare a population structure in two different conditions. And we first looked at both in the immune and the epithelial compartment. In the knockdown condition, we see a dramatic remodeling of the immune compartment within 2 days, but actually the epithelial compartment doesn't change that much under this IL-33 compartment. It's really that first step of IL-33 no longer talking with the immune compartment that causes its dramatic remodeling. But if we look after 3 weeks in the pan-in state, we see that most of the action and most of the change is now in the epithelial compartment right here, and there's many populations that change. We can look and ask, what are the populations that change? Where the blue populations are populations that are depleted, and the red populations are increased in the IL-33 mutated mice. And again, we can label them because we know everything else that's going on in these cells and what modules they express. And we can see that the progenitors are depleted because the cells seem to go forward and then get stuck in the sort of early downstream progenitor, and they don't reach all these sort of gastric and tuft and neuroendocrine states. In fact, the impact of the population is directly proportional to the degree of influence in the IL-33 network that we inferred. So really showing how the computation directly ties in to what we see under perturbation. And to really summarize this little part, we see that a KRAS mutation succeeded in increasing plasticity dramatic in the system by both remodeling the epigenetic net landscape and under inflammation, remodeling the crosstalk with the immune system. I'm going to quickly rush through the story because I want to leave time, and I'm sort of going to skip some slides. But this is another really cool example of plasticity together with Charles Sawyer's computationally led by Joe and a team of brilliant experimentalists. And here again, I always like studying a simple system. We know that there is a drug resistance that is confirmed by loss of P53 and RB. So we could see what happens when we do that in an organoid setting. And here, when TP53 and RB is lost, we can look at the organoid, and the organoid sort of gets ugly and filled in and loses its morphology, and even develops EMT and slithering and really nasty cells. And we could see that this hyperplasia and slithering phenotypes develop whether we perturb the basal or the luminal cells. So we decided to see what really happens. At 2 weeks, the organoids still look good. So we took RNA-seq of what happens 2 weeks when we still have sort of phenotypic structural integrity, what happens at 4 and 8 weeks. And what we really see is that at already 4 weeks, 2 weeks, when these organoids still look good, there's a lot of transcriptional ugliness happening in them. And what this transcriptional ugliness is, and again, we use an approach to classify, is while at week 0, each cell is either basal or luminal and clearly is clustered as a clear subtype, the cells really lose their basal luminal identity and create intermixed phenotypes in between basal and luminal after mutation. And we can sort of quantify plasticity as a degree of mixing. We can even visualize how ugly this looks, where wild type looks really good and you have basal cells, luminal cells, clearly one cell or the other with very few potential paths of differentiation between them. At week 2, you already see multiple mixed states, multiple potential paths between them, even before you completely lose the structural integrity of the organoid. And by week 4, everything is a mess with mostly intermediate states and very few, pure basal and luminal states. Now, we have this metric of plasticity. So again, we can ask what else is going on in the cell. And we see that the top pathways that are associated with this plasticity score is JAK-STAT signaling. We also see FGFR and a lot of good stuff. Here you could see the correlation between JAK-STAT signaling as a sort of transcriptional signature in the cells and this basal and luminal mixing of the cell. Now, this is autocrine signaling. This is an organoid. There's no immune system here. So what's driving JAK-STAT? We developed a method to say that, well, if this is autocrine, we could look at the ligand and the receptor in the same cell and see if the ligand and the receptor are high, when is JAK-STAT signaling also high? So we built a very simple model to say ligand plus receptor should predict JAK-STAT activity. And when it does, to a significant degree, we say this is a potential ligand receptor interaction that's driving this. The other thing is, again, this thing should be different between the wild type and mutant because we know that it's different. So At the top of our lips are our LIF, which doesn't have an inhibitor, so we directly inhibit JAK-STAT and FGF. Combined JAK and FGF inhibition reverted the organoid to its normal, nice, hollow cystic phenotype rather than this, filled in, ugly hyperplastic to a significant degree, where each individual JAK inhibition or FGFR inhibition also significantly improved things, but not quite the degree as the combo. And so really computationally, we could predict what is driving the plasticity and what might revert this plasticity. We see the same phenomena in mice and in clinical human samples. And the real cool thing is that this not only reverts the organoid to its plastic shape, but it also reverts the drug resistance and now makes these drug resistant organoids sensitive to androgen therapy drug. So we could actually revert drug resistance, showing a potential therapeutic opportunity to sort of target this in patients that are likely to get drug resistance to androgen therapy and metastatic prostate cancer. Again, showing the power of how we could use plasticity and understanding what's driving it to really block plasticity in its tracks as a potential approach to study cancer. So we talked a little bit about the causes and consequences of plasticity. Plasticity is driven by mutations and aberrant epigenetics and inflammation. The consequences are nasty Nancy cancer cells with many abilities, and therefore being able to drug plasticity directly and stop and curtail this plasticity is a potent therapeutic approach. So thank you for your attention. And I'd like to again highlight Cassandra, Joe, and Manu in my lab, Dorena, Wouter, Sonia, and Marius in other labs. And thank you very much for your attention.
Thank you so much. Wow, what an amazing tour. Are there some questions for Dr. Pira? Got some claps.
I tried to go slower in the methods part and then really highlight how when you develop good methods, you could really get good stuff out of them when you carefully integrate good methods with good data and a good communication with the biologist.
So feel free to put your question in the chat or to simply turn on your microphone and speak up.
So I see a question from Dylan.
Yep.
So another approach that we're working on, which I didn't talk about today, is attack velocity. I mean, really, I think that for very long-term things, it's very hard for a number of reasons. Our attack velocity algorithm, which sort of says, okay, we can try and use attack-seq to sort of learn a model of transcriptional regulation. And if we have such a model of transcriptional regulation and we see the open peaks and the activity of transcription factors, we could predict where this regulatory module will drive the cell next. And we see that this has longer range transcriptional regulation. You could sort of connect up to a week, maybe. Some of these processes, it's a little bit more robust. Many of the problems with some of these longer things, and particularly to try and connect dots in cancer, is a lot happens and things can happen, twist and turn, and we don't really even observe the intermediates. So I really think that you basically don't have the data for really, really long-term connections unless you really develop the fundamental understanding of cancer progression. But attack velocity, which is again using transcriptional regulation and combining attack-seq and RNA-seq to sort of predict where a cell might go, does have a longer range and is actually more robust than velocity-based, sort of splicing-based velocity. The advantage to splicing-based velocity is you have all the information in one single cell RNA-seq data set and you don't have to bring in additional data.
So question from Guillaume Book. Guillaume, do you want to turn on your microphone?
Sure, I have some lights. I was curious to know about the post-transcriptional changes and whether you're also able to detect that or you explore that in some of your models.
So, it's really nice to, these sort of genomics techniques that allow you to go genome wide and global and easy and get lots of cells and lots of measurements are really nice. I mean, many of the original methods were actually developed for CyTOF, which includes post-transcriptional measurements. We do see the impact of signaling on many transcriptional signatures. I mean, signaling does have impact. And sometimes when you have a little bit of prior knowledge, we can sort of really map and sometimes incorporate known signatures. For example, I incorporated a JAK-STAT signature into my data. I incorporated an FGFR signature into my data. We've used oncogenic KRAS, ERK signature, MEK-ERK signature. So through signatures, because a lot of these post-transcriptional changes have some signature in the data, you can sort of think about what signaling might be active in a cell, assuming that your signatures are derived in the right context, because signatures are very context specific, and a signature you'll get in some stupid cell line for a different cancer are not necessarily representative of how your cells of interest are going to behave. But yeah, post-transcriptional is a whole new world, and our technologies to really understand it are still very limited.
So there's a question from one of the students in the classroom. You calculated absorption probabilities for different terminal sulfates. Did you calculate conditional mean first passage times?
So again, this was, what we assumed here to make our life a little bit easier, and it's also slightly different between Palantir and CellRank. But here we really use the closed form solution. So we assume that our observed states have no outgoing edges. And therefore we basically ran the Markov chain to infinity. And basically each cell, the walk stops when it reaches an absorbing state and cannot leave it. One has to say that these walks are not necessarily representative of actual time. They're sort of more potential paths through all these different states and transitions. But we know that many of these paths and the lengths of these paths and probability of these paths do not correlate to any actual time.
There's a question from Abu Kumaran. Thanks for the great lecture. Two questions. What algorithms or methods do you propose to investigate bi-directionality in sulfate decisions? And have you performed analyses on isolated cancer stem cells? Would be question two.
So one of the problems with, again, some of the organoids is sort of like cancer stem cells. One of the big problems of cancer stem cells or any sort of isolated analysis is that we know, and I just showed you in the PDAC example, how important the cell-cell interactions are. I mean, we've cured cancer in a cell line and in a dish a million times over. We are actually using some interesting co-culturing experiments where at least we can, I really like it when there's at least one interaction at play and less looking at completely isolated stem cell. Even though, again, the organoid situation, one could sort of, the prostate cells were stem-like in their origin. In terms of bidirectionality, again, I think this concept of attack velocity and regulatory module to sort of say what is the potential to go here and there is a very powerful approach to model bidirectionality, because you could sort of predict. And we saw in the sort of work that I showed you in pancreas, where each cell has the ability and the probability to go in multiple very different directions. So the current attack velocity, not yet published, but at least completed algorithm, does not model bidirectionality well, because the more complex your model is, the less robust it is. But now we're really trying to understand the system so we can, in a robust way, allow the cell to go in any which way based on the sort of attack-driven regulatory model.
Thank you.
But that's still very much work in progress. There's always a very, very big trade-off between flexibility and robustness, and the key is always to see how realistic you can be without, you know, overfeeding to noise, and it's always striking that balance and understanding the system more to constrain it more so you can get increasingly realistic models nevertheless.
Are there any other questions?
I have a couple, but I'll save them for the
We'll save them for later, Selenia, great. I'm going to take my prerogative and say, you know, you started off with nice Markov models, which are going to be unidirectional, right? So is part of the problem with bidirectionality the fact that you're going to have to throw that out the window or break some of the assumptions that are lying there?
Well, the question is, how do you robustly, I mean, Markov models, as you said, are bidirectional. And at the end of the day, you know, if we sort of look here at this model right here.
We don't see your slides anymore, I'm afraid.
I'm going to share it in a moment once I find it. It's
Okay.
just quicker for me to scroll. I know I'm not sharing yet here. This is it. It's just, okay. So in this slide right here, we do have probability in all directions.
Mm.
But because there's only one velocity, it's really dominated by one direction. So currently, if I look at the cell rank, there's you can go in all directions, and there is a non-negative probability, non-zero probability to go to any neighbor, but it's really dominated with its alignment with the velocity direction. Our current attack velocity algorithm, again, is a linear model that, again, creates a dominant, most likely direction. And while this velocity is computed based on attack seek rather than based on splicing, we have a similar dynamics in terms of what's happening. Now, when we try and make a sort of softer model that sort of says, what's my probability of going any which way? We really lose robustness. And when I say robustness, I'm not even talking about biological correctness. I'm talking about perturb the data a bit and get the same answers. Like my first, my first test for any algorithm is it going to give me the same algorithm if I, do some sampling with replacement, or if I add a little bit of noise to it? Because if it's not robust, then it's bullshit. And so to release the sort of hard constraint as this is the most likely direction, and I'm pretty confident about this, so this is robust, into let me give you a probability for any which way direction, and it will be different if I slightly perturb the data, is where we're working right now.
Thank you. A last chance for questions. I'd like to remind all the QLS students that they can stay, and Dr. Peer will be taking some time to chat with you. Okay, well then, in that case, I'm going to say a very sincere thank you for a wonderful presentation covering all kinds of thought-provoking ideas for future research and directions. Thank you so much.
It was fun. Good questions.